All right, so please take your seats. I'm delighted to open session one of the conference. The first speaker is Professor Dan Peer from Life Sciences. He changed hats from an administrator to a scientist. And he will tell us about the design of novel mRNA vaccines from viruses to bacteria. Professor Peer, please.
Thank you. Okay, thank you so much for the kind invitation, and I want to apologize that I will have to leave after my talk. Anyhow, it's fun to talk about science. So, central dogma of biology, as we all know it, that we have the genetic code, the RNA, and then proteins. And as you can see here, the RNA is being investigated over the years from Mr. Bean, and that's been 22 years ago, this image, that basically needs a better template, somebody to copy from. And even the mother of dragon already showed us the difference between double-strand DNA and a single-strand RNA. And we have been interested in this field for a lot of years, and from a therapeutic standpoint and vaccine standpoint, this is the area of the RNA. In the past few years, you can do many things. You can silence genes, you can upregulate genes or activate them, you can edit them, and hopefully in the near future, you'll be able to replace them. So we've been engaged in doing some pioneering work, both at the level of silencing. We were the first, our lab was the first to show we can deliver siRNAs to leukocytes in vivo. We're the first to show we can deliver messenger RNA in a cell-specific manner in an animal, and that was much before COVID. And we're the first to show genome editing in cancer. And today I'm going to tell you a little bit about the work that was published a year and a half ago, but is progressing very quickly to the clinic and involve bacterial vaccines using mRNA. So the challenge in RNA therapeutics, but also in vaccines, is twofold. The RNA itself, which I will not talk about it today, but it's a huge challenge. I'm actually going to mention it a little bit, and the delivery. And you need an appropriate delivery system, and it has different requirements. It needs efficient entrapment. It needs to evade clearance mechanism, to avoid toxicity and immune activation. The carrier needs to spill out or spill in, whatever you want to think about this, its payload into the cell. Could be internalized or not, and the RNA must be released. And we need specificity. And the candidate that is now very much into place are lipid nanoparticles with the RNA inside. And this strategy is being engaged very quickly. The scale-up stories are very quickly done using currently microfluidics mixers. And there are a few compounds that are involved. There are mixtures of 4 or 5 lipids, mostly cholesterol, helper lipids like phosphatidylcholine, some PEG lipids, and probably the most important one is the ionizable aminolipids, a pH-sensitive lipid. There are lots of advantages of using lipid nanoparticles, but also some disadvantages. The advantages are they are biocompatible to the biological system. They're fully degradable. They protect the nucleic acids, they increase the stability in circulation, could also increase the therapeutic effect, and some of them are clinically approved. For example, the first example is an siRNA from 2018, was FDA approved, the Onpattro or patisiran. This is basically an siRNA against TTR amyloidosis. And the 2 vaccines, the Moderna and BioNTech vaccines, or I would say the BioNTech-Pfizer vaccines that are approved from the emergency approval of December 2020. In our lab, we are interested in utilizing different approaches, and we do everything from biology into the delivery. We synthesize lipids. We have the largest lipid library probably worldwide right now with more than 100,000 lipids. 7 of them are being clinically tested as we speak. 3 phase 3 trials in cancer. The rest are cancer vaccines and vaccines for infectious disease. And behind the scene was a former postdoc of mine, Srinivas Rameshetty, that basically took in 2011, 2012, the structure of the DLIM family, DLIM MC3 DMA, that a few years later, about 6 years later, was approved by the FDA, and start to engineer it or create a reverse engineering, basically looking at the head group, the linkers, and the tails, and came up with a lot of families of lipids. And we're very lucky that we could patent this as well. And so Srinivas became a very rich guy. So he's now in Oxford, having a lot of fun. Microfluidics, as I mentioned, is also very important. It makes our life very easy. And we're also involved in the creation of a company named Precision Nanosystem. Exactly 12 years ago, the company was sold two and a half years ago to Danaher, and everyone was very happy. So what can we do? We can characterize those structures, those LNPs, and we can make them even better. So this is how we do it. We screen, we basically start from synthesis, and then we do a lot of formulation sciences, and we come up with an excellent candidate. And we can tune immunogenicity for vaccine. We can make them more immunogenic, where if we want to do therapeutics, we want to do much less immunogenic. And this is a recent example of more lipids, more structures, etc. Somewhere in 2020, I was approached by Ofer Cohen from the IIBR saying, we need to do something in COVID, let's work together. And we decided to basically utilize a student, a joint student that worked in the IIBR, and basically gave him a project. And so we designed RBD structure with human FC, mRNA construct, and we challenge human ACE2 transgenic mice against a little SARS-CoV-2 challenges. And this is Uri Elias's work that graduated in April with his PhD. And this was a very fruitful collaboration. As you can see from the data, we get very nice protection in very aggressive challenges. And Uri published very nice 2 papers. So everyone was doing basically COVID. And also we were asked to write what they called a fast response for highly mutated pathogen commentary in Nature Nanotechnology. And we started writing it in 2021. It was published a year and a half later, and you can see how editors sometimes take their time. So the best one was Omicron 1.1.529. When this paper was published, Omicron 5 was already around, so it was less relevant, but at least it gave us a little bit of an opportunity to discuss what are the strategies and what do we need. And we do need a lot of things. But looking at the history of pandemics, it becomes very realistic to see that compared to COVID, there have been other pandemics. And probably the most interesting one in terms of size and probably effects was the Black Death caused by bubonic plague. And so we look at the numbers and of course the years and the technologies around, and basically decided that we need to work with bacteria as well. So this is one of probably hundreds of drawings somewhere between the 14th and the 16th centuries that describe the plague. I have another one which is even more nicer and relevant to Israel. And so looking at the gold rush time for mRNA vaccine, mostly were looking at viral pathogens and a little bit about cancer vaccines. And there were very limited data on this platform applicability against bacteria. There is also another issue that is an emerging issue, which is antibiotic resistant, that necessitate alternative countermeasures. So according to the World Health Organization, in 2015, we will all probably be affected by the AMR, by AMR, so antimicrobial resistance. And it's better to start and develop something now if we don't want to die from any bacterial resistance based on antibiotics. So around 2020, there was a very small conference organized by BioNTech and Moderna, and I had the pleasure of giving some talks, and I had an idea of using a bacterial pathogen, and so I talked to Kati Carico, then just a lady from BioNTech who I met in 2013 when she moved there from UPenn, and basically I asked her about her opinion. So I asked a naive question, bacterial mRNA vaccine, and she said probably won't work. And I said, why? And she said, because bacteria brings their own proteins. They are much more sophisticated. Look at their surfaces, tons of sugars, something that generating a really good immunogens will be a huge challenge. So, challenge accepted. And these are some of my lab mates, power girls, that although it looks very similar, it was 1.5 years later. Okay, so you can see that was a conference in Berlin. That was also in Berlin, but that was only for 25 people, and that already 600 people. So this is the differences, and we accepted the challenge. And here the IIBR also gave us a very good potential bacteria, Yersinia pestis. So it's a gram-negative bacteria, the theological agents of plague, highly infectious and lethal, and millions of deaths throughout history. Several hundreds are still dying annually. It's a bioweapon, potentially a tier 1 select agent. No approved vaccines, and basically can be treated with antibiotics, but there are strains that are highly antibiotic resistant. And there are two major performances of this, the bubonic and the pneumonic one. And we started with the bubonic one. This is Nicolas Poussin in the Louvre, the plague in Ashdod. Okay, so just to show that, look at Ashdod. Okay, very nice. At least in the eyes of Nicolas Poussin, who had never been in Israel or in Ashdod. And looks very Roman, I would say. And look at the people, but this is the plague in Ashdod, and this is a very nice picture that is hanged in the Louvre. So when you're there, hopefully next time, go and see this. It's really interesting. But still, if we look at the plague, there are different countries still affected. Mostly countries you probably less visit unless you want to count also US, the western part of it. And basically, every year, about 300 to 600 deaths from outbreaks of plague. So the mission that Uri and Ido got is to try and create an mRNA LNP vaccine against this highly bacterial pathogen, and using one of the strands that is highly antibiotic resistant with the help of Emi Mahmoud and Ofer Cohen, and also with Inon Levy and other people that really made a lot of help. Just to give you a sense, the bubonic plague, normally if it's not treated in time with antibiotics, is 50 to 60% mortality, and the pneumonic one almost 100%, and it transfers in droplets. So we decided to start with a low-hanging fruit, although it became not low at all. And so Choosing and selecting the best immunogen, we had a lot of discussion what will be the best option. And F1 is a chaperone in the pestis, also called CAF1. And V is, again, these are two major protective antigen for pestis that are known for quite a lot of years. So we decided to start by generating in IVT an F1 structure that will be used for vaccination. And this is a classical construct with a cap, with a signal peptide, with a sequence and a poly-A tail, and that was straightforward. And so we used a lipid from our library that is now being used in phase 2 clinical trials by BioNTech in their cancer neoantigen program and personalized vaccine project. So we knew it's actually a very good lipid. And you can see also from all the parameter, physicochemical parameter, that the encapsulation efficiency of RNA is very high, that it's not really important which control we entrap from an mRNA standpoint. The size is very good. They're looking very cute, those round particles. And it's also expressed nicely in HeLa cells. So we decided that it's good enough to start. And so these trials, these in vivo trials were done at the IIBR. First, they injected 5 microgram of the mRNA with LNP, then boost, another boost, and then the idea was that we'll create a challenge. You can see the bleeding after 2 weeks and 4 weeks. Surprise, surprise, we didn't get, after 3 bleeding, any titer of antibodies. And that was a bit problematic. So Kati Kariko was probably correct. And we had a control, which is basically the recombinant protein with alum, and you can see it gets nice antibodies. But we did get some interferon gamma response, which was not bad. So we do get a cellular response, but we don't get protection from antibody production, which means that we basically will not challenge the animals. It's a waste of time. Okay. So then we decided to play a little bit with the mRNA, with the construct. So we knew that it's not a delivery issue because those lipids are immunogenic. And we know that in humans, they're giving very good response. What happens in mouse? So here we played with GC, so putting GC rich and repeated the experiment, already started to get some kind of antibodies after a second boost. And also in a challenge trial, we started to see some protection. Not great, but 50%. In this bacteria, you can see that the survival of animals is, they all die on day 5, so you get nice protection. And we decided to continue with this. So we added the trick of human FC and the high GC contact, and also we took out the signal peptide. There are some papers, a bit old, showing that if you take out, you create a delta signal peptide, you get a better immunogen. So we tried it again. This time, we already got, even in the first bleed, we got very nice, before the boost, very nice starting point of responses. So we did a challenge trial, and you can see 100% protection. And that was very encouraging because this dramatic effect, basically taking out the signal peptides, gives you very nice protection. And of course, human FC looks very nice. So we got greedy. And we decided that maybe we should just do a prime and then challenge. We did a few bleeding, but only a single prime. And surprise, surprise, in the human FC portion, you get 100% protection. and you get it already from day 3 to 5. Are we challenging immunology? So I presented this a week ago at the Jenner Institute in Oxford. And all the immunologists, some of them are very famous, basically said you should dig into the immunology because something here is strange, right? Because we all know in textbooks that to create antibodies takes time when you have a first-time exposure to immunogen. In mice, it was documented that the first response that you get effective antibodies are 7 days. Here we get actually effective antibodies in titer at 3 days. And I would say more comfortably 4.5 days. So there is a different immunology here that we need to understand. And as you probably know, I'm not an immunologist. So we're now collaborating with several groups, also with the IIBR, and outside the IIBR as well, to try and understand how effective. This is a highly reproducible results. It happens all the time. Just to give you a sense, we're already in the process of making GMP production of this. Hopefully very soon, we will be able to test in human just the level of antibodies, how much those are there and how quick they are. There is still a lot of unknown, but we are very optimistic. So I know I'm a bit over time, I think. No, I have a few more minutes. So just to summarize, we understood that the mRNA payload optimization is crucial for successful design of this potent mRNA LNP, specifically against the pestis, but probably for other pathogens. We're also working on other bacteria. The optimized construct here basically yielded a robust humoral response, and we are super interested to see if we also get a T-cell response that will create memory over time, because this is right now the major problem in this field. And this work was the first to utilize an mRNA LNP platform against a highly lethal bacteria, basically a proof of concept. And this provides basically new avenues for combating antibiotic-resistant bacteria. When the paper was published, I was asked by the Gates Foundation to give a talk at their scientific advisory board. And they were intrigued from the fact they're not interested in plague, you can imagine, but they are interested in other bacteria. So I think that there is a lot of opportunities here for understanding and doing and improve versions of vaccines against antibiotic-resistant strength. And at the end, I want to thank my group, talented people who work there, people who help us in other projects as well, funding agency and our collaborations, and thank you all for listening. Yes.
Many figures. Thank you, Professor Per. Natalia?
Hi, amazing talk. I was wondering if you had checked for pre-existing antibodies in the mice.
For plague?
I mean, for your antigen, because what happens is
Yes.
when you prime, you might be boosting something.
Zero. Naive mice. Never been exposed to this. I want to mention that the challenge trial, of course, were done in IIBR, in a BL4 lab. We don't have, it's not even 3, so we don't have access to this. We don't do it academically. But I think it's very interesting. I can also give you the strength, a specific strength if you're interested, because there are a few options, but it's also written in the paper.
But you might be boosting something that's not strong enough to protect, but cross-reactive.
Yeah. So these are the things that we are checking now. So we want to see, but again, as far as we saw for the F1, There are no antibodies at time 0. So nothing there. Also, they don't seem to have any T cell activity. Probably, I mean, not an interferon before we do this. We also had controls with the LNP that is empty, all the controls. And with them we get 0. Like there is nothing. But it's interesting. Right. Right. Right, right. Right. So one of our tasks now is to find both lipids and construct that will have higher T cell activity. We also look at B memories because at the end you want to have memory on this. But right now, what we have seen and what is commercially available, they have no memory. And there is also some adverse effect that probably create a more complex system to the immune system. So I think that there is, again, a lot of room to understand all the parameters. And now it's a time of, we were in the hype. Now we kind of at plateau. In the plateau, hopefully we'll do a better science. But I think running very fast was the necessity of the timing of the period, yeah? And everything else is now it's the time to study it. And when you study it, you get amazing things. For example, I don't know if you know, I mean, it's known already in the field, but if you remember the first COVID vaccines, both Moderna and BioNTech says on the vial, discard room temperature after 6 hours. You remember that? It was written on the vial. You also remember that people who were vaccinated, some of them became sick with COVID. No, because they didn't get anything from the vaccine. So now the data is out, okay, and in the conferences and in some papers, they're starting to talk about thermostability of the vaccines. And what we learned is kind of not surprising, right? If you remember, Moderna vaccinated 100, the dose was 100 microgram. BioNTech, Pfizer was 30 microgram. But all of them, after 6 hours, was 0 microgram. It was completely degraded. So people who got vaccinated in the vial, and they were number 4 or 5, and the vial was, not like in Israel that everyone was vaccinated like crazy, but in many places in the world, it just sat for a few hours. The people who got the last dose were probably not protected at all. Placebo, exactly. And some of them probably were immunized, others not. So this is something that did not happen in the second batch. So second batch appeared about 6 months later. The people who got the second batch already, it was tolerated very well because the second batch was thermostable for at least 48 hours in room temperature. And they took off the label of it. And the third one was 2 weeks.
I'll ask 2 brief questions. So, first of all, you're still looking at it, but is there evidence, were some of these mice followed to see persistence of protection?
There is no persistence of protection for a very long time.
And the second question may be the fact that, so I don't know about lipid, that was lipid 17, right?
No.
Oh, never mind, but anyway.
40.
Well, potato.
Not really, they're all different.
But, no, concerning biodistribution, so most LNPs, if no action is taken, go to the liver and the spleen. So may it be that the particles themselves act as super adjuvants by going directly to the spleen and starting?
Oh, of course, these are injected IM. They go to the spleen, mostly. The liver also, but less. Yeah. But we see it in humans.
Thank you very much, Professor Peyer.
Thank you.